Okay, thank you very much, Jordan, for inviting me to this wonderful series of webinars. And also thanks to everyone who have attended today. It's a great attendance today. Um, because I need to dip away immediately after my presentation. So I'll be very pleased if you kindly send your questions to my email address that I'll be giving at the end of this presentation. I'll be talking about the comparative analysis of leaf physiology and anatomy and dephenotyping in wild and cultivated tomatoes to investigate differences in photosynthetic performance under varying light and CO2 levels and how these relate to stomatal physiology. Okay, so I'll directly go to the contents. Uh, we have studied two species of tomato, which one of them is called Lea and another is Lost. Lea is Solanum lycopersicum, which is modern tomato inbred that is suited for processing tomato industry. And Lost is it's a green fruited drought tolerant wild relative of Lea. Because these two species have differences in their morphology and sensitivity to environmental stresses, and also because the interspecific backcross inbred lines that have been generated from the cross between these two species facilitate mapping of very important agronomic traits like stress and yield, uh, and also the causal genes. We selected these two genota species for this study. So the main thing that we'll be talking about today is the photosynthesis, the differences in the photosynthetic capacity between these two species. However, before we go to that, I want to say that I want to share this thing that while we were growing these two tomato species, we observed a very interesting trend in their growth. Initially, if you can see, this is the lost wild species. It was very small in height, and the cultivated species was very like it was higher in higher. It was and the growth rate was also very. Three leaf area was also higher as compared to the wild species. However, with time, this wild species overcome this cultivated species in the plant height. And and this on the on the right side, this is when they were setting the fruits. And this wild species also it takes a lot of time to set the fruits. This basically this lost the wild species growth pattern while it overtakes Leah in height around 23 days. This is shown also in this graph. Like initially, the 3D leaf area in the Leah was is almost higher throughout the measurement period, while as in the case of height measurements, it first is lesser than the cultivated species, but around 23 days after transplanting to the pots, it the wild species started overtaking Leah in plant height. This growth pattern, while the wild species overtakes Leah in height around 23 days after transplanting, suggests a adaptive strategy that balances water conservation and the light capture, and bears similarities to the conservate and bears similarity to the growth strategy observed in some desert plants, where growth is initially conservative but can rapidly accelerate vertical growth to enhance the light capture. Now, coming to the main topic is comparing their photosynthesis. So, as we know that, and also for those who might not be very much aware about the photosynthesis in the audience today, so this is for them. It mainly comprises of the two processes: electron transport that happens across across these components in the electron transport chain, and the CO2 assimilation. We'd first talk about the electron transport between these two, rate of electron transport between these two species and how it differs. Um, for that, we used for the characterization of this photosynthetic electron transport between these two species. We used dual class. is an instrument which uses two 4 wavelength pairs in the near infrared region to simultaneously describe to determine the oxidation and reduction chains of very important components in the electron transport chain, which are plastocyanin of P700 and ferroxide. These are the light response curves of electron transport rates at photosystem 2 and photosystem 1. And we saw that, as we can see, that with increasing light intensities, the electron transport rates at both photosystem 2 and photosystem 1 increased in case of lost. Not only the electron transport rate, but also the oxidation of the plastocyanin P700 and the reduction of the ferredoxin. So in the oxidation of the plastocyanin and the P700 were higher in case of lost, and while as the reduction was lesser in case of the lost with increasing light intensities. So these results suggest that these components are always in the oxidized form, like they are always in the deficiency of the electrons, because the processes downstream of these components, they always take the electrons from them. That means it might be the sink, like the Calvin cycle, that's taking the electrons from these centers, and they're always oxidized. And that means the electron transport is very efficient in this wild species. So these, besides these electron transports and oxidation redox potential of the different components, we also wanted to know the photosynthetic yield at the two photosystems. And we can see that although the yield, that's how efficient the plant is to convert the photosynthesis, to convert the light energy into the chemical energy or the biomass. It decreases with increasing light intensities. However, it's mostly higher in most of the light intensities. It's higher in case of the wild species than the cultivated ones. From the non-photochemical quenching results were a bit of counterintuitive, because non-photochemical quenching is like when the plant dissipates the excess energy as in the form of heat. So you would expect, if you have higher yield, if you have higher efficiency to convert the light energy into the chemical energy, the NPQ should be lesser. It should be dissipating less energy, like wasting less energy. But like we thought it could be because this wild species grows in South American arid regions, where it is very hot and very high. There's very high light intensities, so it has to protect itself from high light intensities and reactive oxygen species that are generated due to this high light stress. So it means that this wild species lost is both efficient in converting light energy into the chemical energy. Not only that, but simultaneously it also protects itself from photo-oxidative damage and photo inhibition. So these were some results from the electron transport rate side. Now coming to the carbon assimilation rate in response to different CO2 and different light levels between the two tomato species. So we use that. Most of you might be aware about this instrument that's called LICOR 6800 that we used to get these data. So the gas exchange that was done using the LICOR 6800, the carbon assimilation under different CO2 levels and the light levels was higher in case of the wild species than the cultivated species. The higher assimilation rate and higher CO2 levels, that means it has higher maximum rate of carboxylation as allowed by the Rubisco in the wild species, and indicating a greater efficiency to fix CO2 at high CO2 levels, which has implications for the global climate change, as CO2 levels are expected to increase with time. Similarly, in these light response curves, we saw the photosynthetic assimilation also increase in this wild species. and it also indicates that it has higher light use efficiency as compared to the cultivated species. Central to the carbon assimilation is the Rubisco enzyme, as we all know. So we wanted to know how the Rubisco activity differs between the two species. Both the Rubisco initial activity, which is the activity of Rubisco immediately after extraction, and the Rubisco total activity, which is the maximum carboxylation potential of Rubisco after it is incubated in high concentrations of CO2 and magnesium. Both these activities were higher in case of the wild species. And not only the Rubisco activity, but the Rubisco content were also higher in case of the wild species. Also, the ratio of the Rubisco to the total soluble proteins and the ratio of Rubisco to the total chlorophyll content were also higher in case of the wild species. All these results, they complement the high carboxylate, high carbon assimilation in the wild species. From the stomata point of view, we wanted to understand how these differences in the photosynthesis between the two species are explained. So we determined we measured the stomatal density and pore size on the baxial and adaxial leaf surfaces in these two tomato species. And we observed that as compared to the Lea, the two surfaces in case of this wild species had similar stomatal density from the two surfaces, whereas we did not observe any difference in the pore length from the two surfaces between the two species. This ampistomity, that's the similar number of stomata on the two surfaces in the wild species, is the need of this plant genotype because it's because of thick leaves that I'll be talking about later. To ensure, because if a plant has a thicker leaves, the gas has to the CO2 has to go to the deeper layers, deeper cell layers of the leaf. So to measure again, sorry, to understand how this difference in the stomatal density from the two surfaces of the leaf, they translate to the differences in the gas exchange between these two surfaces. We wanted to analyze the gas exchange from the two surfaces simultaneously but independently. So for this, we cannot use the Lycor because Lycor 6800 it gives us the gas exchange from leaf as a whole. Well, as here we wanted to determine the gas exchange from each surface independently. So this is a kind of gas exchange system that a lab has already developed, where you can do these kinds of gas exchange systems from the two surfaces simultaneously but independently. Okay, so this is again the results of the stomatal density that I have shown previously. So to understand how this different stomatal density translates to the stomatal conductance in the two species, it was perfectly corroborated because Lea, which had higher stomatal density on the abaxial surface, has stomatal conductance on the abaxial surface than its adaxial surface, which had the lower stomatal density. Whereas in case of the wild species lost, which had the similar stomatal density on the two surfaces, the stomatal conductance was also similar on the two surfaces. Likewise, in case of Lea, the carbon assimilation was also higher on the baxial surface than the adaxial surface, corroborating with its stomatal density. But we were surprised to observe these things, these results in wild species, where we found, despite its same stomatal density on the two surfaces, the carbon assimilation on the abaxial surface was significantly higher. And we initially found that this could be something technically, could be a technical mistake. So we repeated it a couple of times, but the results were the same. Okay, then we wanted to know what the underlying factors could be explaining this. So for that, we measured the leaf thickness, leaf mass per area, and also stomatal structure, mesophyll structure in the two species. And it was observed that, and we did this, by the way, with an instrument called multispec. And we observed that both leaf thickness and leaf mass per area was higher in case of lost. And we also did the microtomy. Okay, yeah, sorry. This higher leaf thickness could indicate that the concentration of metabolites in the wild species could be, and cell metabolites and the photosynthetic proteins could be higher in this wild species. The microtomy results showed that as compared to the Lea, this cultivated species, the wild species had larger and highly packed palisade mesophyll cells, like these palisades. But more important than this was, it had more spongy mesophyll cell density in as compared to its cultivated counterpart. So this could explain this because spongy mesophyll is towards the abaxial surface, which could explain its higher photosynthetic capacity from the abaxial surface. Not only that, so this higher mesophyll cell density might also explain that it might have a higher number of mesophyll chloroplasts and high chlorophyll content. So, in order to know that, we measure the chlorophyll content from the in the two species and we found that both chlorophyll A, chlorophyll B, as well as their ratio in lost was higher as compared to the cultivated species. Next, we also wanted to know if what about the guard cell chloroplasts, which are in the chloroplasts, which are not in the mesophyll but in the guard cells. Uh, it was interestingly found that the abaxial surface in the two tomato species had higher density of guard cell chloroplasts, but the difference between the two surfaces was higher in case of wild species. But more important observation than these average number of chloroplasts in the guard cells was the arrangement of these chloroplasts in the guard cells. Guard cells, as we can see here, guard cells in the lost are fully populated with chloroplasts, while as in case of Lea, they are sparsely distributed. This difference in the chloroplast arrangement and distribution could affect the way the light is captured and used for photosynthesis, and it can also affect the way how plants manage gas exchange and water loss through stomata. Lastly, we wanted to know if this high photosynthetic capacity trait in the wild species lost could be transferred to the Lea through grafting, and to see if there's anything in the roots of the wild species that could be contributing that could contribute to the that could contribute or transfer this trait to the cultivated species. So this is an example where we grafted the cultivated species on the wild species, and here we grafted the wild species on the cultivated species, and these are the different combinations. So we firstly determined the stomatal density on the abaxial and axial surfaces on these non-grafted and self-grafted and reciprocally grafted plants. So these here I have shown the grafting combinations, like non-grafted ones which were not grafted at all, and self-grafted means where Lea was grafted on Lea and the lost was grafted on lost, and reciprocally grafted where they were grafted on each other. We were most interested in this grafting combination where we grafted cultivated species on the wild species, and we wanted to know how the stomatal density compares with respect to the non grafted and the self grafted layer. So although in all these combinations, Leah, non-grafted Leah, self-grafted Leah, as well as the Leah that was grafted on the lost, the stomatal density was higher on the abaxial surface than adaxial surface, and it was also true in case of these grafting combinations. But we can see that the difference between the two surfaces was decreased in these grafting combinations. So that means the lost rootstock could be inducing this equal stomatal density trait in this grafted cultivated scion. So this is the so we wanted to know if how this difference in the stomatal density could be translated into the photosynthetic performance. So we saw that although in these grafting combinations, still it was the loss which had highest stomatal this carbon assimilation rates, whether it is ACI or the AQ curves. But we were most again here we were most interesting to analyze the Leah that was grafted on the lost as compared to the non-grafted Leah or the Leah grafted on itself. The photosynthetic capacity of the Leah that was grafted on the lost was higher. So lost rootstock, we could say that it improves photosynthesis in Leah science compared to the self and non-self grafted Leah. And it might suggest that there could be some factors, some hormones, some metabolites, or some mobile RNAs that move from the rootstock of the lost to the sign of the cultivated species. Not only it indicates something is from the roots of the wild species, but because if you see here the lost that was grafted on the cultivated species also had a higher photosynthetic efficiency than all the combination of the layer that was either grafted itself or grafted on lost. So it is a combination of both, where there could be something in the leaf of the wild species, and also could be also something in the roots that's imparting this photosynthetic higher photosynthetic trait in the cultivated species. So we concluded that the photosynthetic advantage of lost is there, and it also opens the avenues to find what metabolites are transported across the grafting junction that could further shed light on which genes could those metabolites be transcribing, and they could be used for the generating the transgenic plants. And it also opens the avenues for enhancing the tomato productivity through trait transfer. And lastly, I would like to thank Capitalize Project from the European Commission, who funded this work, and also the University of Essex, and all those members who participated in this work, in this project. Thank you very much. And this is my email address. And as I requested in the beginning, that I need to directly go to the another meeting. And if there are any other questions, please feel free to forward them to this email address, and I'll be very happy to answer them. Thank you very much.